Hi everyone, my name's Eileen Regan. I'm the CCO at Examen, and it's our pleasure to welcome you again to the third webinar in our 2022 Male Fertility Series. So as many of you know, these webinars take place on a monthly basis, and we're joined by an expert in the field of fertility treatment or therapies. The event's free, open to everyone, so we have a wide range of participants from academics, healthcare professionals and those in the wellness, as well as some of the men, couples or individuals who are on their own fertility journey as well. Just to say that the views presented in these webinars are the views of our speaker and not necessarily of the company Examen. So we're absolutely delighted to have you all join us today. And let me introduce you to our speakers. So this month we are joined by Professor Dolores Lamb. Professor Lamb joined Weill Cornell Medicine on March 1st, 2018 as Vice Chair for Research in the Department of Urology and Director of the Centre for Reproductive Genomics. She holds the Robert S. Dow Professorship of Urology, and she maintains an active presence in both the academic and research communities at Weill Cornell Medicine. Dr. Lamb is an investigator in the field of urology, male infertility, steroid hormone action, prostate cancer, and genitourinary birth defects. Her experience is unique as she has an extensive background in both the clinical diagnostics and the basic science arena in men's health. She's a highly recognized NIH-funded researcher whose areas of investigation have focused on the genetics of male infertility, the genomics of genitourinary birth defects, steroid-regulated growth of male reproductive tumors, and other areas of benign urological research. Our chair today is Professor Shana Lewis, who is the founder and CEO of Examen and an honorary professor at Queen's University Belfast, where for the past 25 years, she's dedicated her research into novel tests for male infertility. She is currently an executive member of the Association of Reproduction and Clinical Scientists and chair of its Scientific Advisory Committee. Professor Lewis has published over 100 full papers, as well as numerous book chapters and reviews. She has been past chair of the British Andrology Society, UK National Representative and chair of the Andrology Special Interest Group of ESHRA, and executive member of the British Fertility Society, and was also a founding member and vice chair of the Irish Fertility Society. Professor Lewis's research has led to the formation of the company Examen, where we market the Exact test, which is now the market leader for sperm DNA damage testing in the UK and Ireland. So thank you to both of our chair and our speaker. We're delighted and looking forward to hearing you speak. Before I hand over to Professor Lewis, I just want to run you through some housekeeping. So can I ask you please to pose questions throughout the talks using the Q&A panel, not the chat function. So if you do, you also have the opportunity in the Q&A to rate questions, and Professor Lewis will then share the questions during the session. If you experience any technical issues, please use the chat function and we'll do our very best to help you. Attendees will be muted throughout and videos enabled just for our speakers. All the events in our series are recorded and are available on the Examen website. If you go to the events page, you'll be able to see those probably tomorrow. And also you can watch any events you may have missed. So give us about 24 hours and we'll get the webinar online for you. One CPD point will also be awarded for the attendees for the event, and we'll circulate a certificate in the following days after the event. And if you would be so kind as just to rate the session at the end, it gives us a good idea of feedback that we can pass on to our speakers and hopefully provide some more great webinars for you in the future. So please let me hand over to our chair, Professor Shana Lewis, to start the session.
So we're going to be talking about the changing face of male fertility. And I hope many of you are saying, well, it's about time, and it absolutely is about time. Male fertility has been ignored for so long, but things are certainly changing. And Professor Lamb is one of the people who has been instigating many of those changes. And it's going to be really exciting to hear what she has to say to us this evening. So as you probably know, the purpose of our webinars is to raise the awareness, of male infertility, and to provide men with knowledge and advanced testing, because that's something that they haven't had for a very long time. And really the purpose of our whole spin-out company from the university is to help more men become dads. Our mission and our purpose is to demonstrate that couple care can improve live birth rates. Also, that optimizing sperm health before treatment can improve fertility outcomes. And we have a very, very big, a big mission and a big purpose and a big objective, and that's to help 1 million men along their way to make those men who want to become dads able to do so. Now, what I thought I would tell you just a moment or two this evening about was some of the things that we have done and the evidence base for our test. Now, the test is based on 25 years of research into male fertility. And during that time, as you can imagine, we have looked at quite a number of different areas. One of them is prescription drugs. And we looked particularly at Viagra, which of course was a hugely popular drug when it first came out. And we looked at the effects of it on sperm function, sperm motility, and sperm acrosome reactions. The second area that we looked at were cannabinoids, and I wish someone else would just pick this up and run with it, because I think this is a very, very exciting area, because we have a hugely complicated endocannabinoid system in our bodies. And we found that there were huge differences in the agonists, the antagonists, the receptors in the sperm of fertile men compared to infertile men. We looked at motility enhancers for men who had slow motility. We looked at diabetes and found how men with diabetes had a lot of oxidative stress in their blood and a lot of oxidative stress in their semen, poor DNA quality. We looked at vasectomy reversals and their effects on sperm quality. And of course, one of the biggest things we've been looking at recently have been men and miscarriage and the male contribution of poor sperm quality to recurrent pregnancy loss. And at the moment, we're looking at DNA damage and unexplained or idiopathic infertility. Now, as well as looking at research into men's sperm, we've also been looking at the need for research into men themselves, infertile men, and what can cause their infertility. So because we have this company, we're able to help collaborating clinics to do research. And one of the reasons we set up the company was so that we would be able to fund research, because it's such a difficult area to fund. So we've been looking at varicoceles and when they're repaired, what the impact is on sperm parameters and also on ART outcomes. And that's very exciting. And a paper will be coming out on that soon. We've looked at infection, and we've also looked at the microbiome. And for those who don't know what the microbiome is, you'll be hearing all about it in the next few years, because it's the next big thing. The microbiome is really a little army of microbes, which actually help us to stay alive, and they do all sorts of things to help us. There are good ones and bad ones, and it's just a question of seeing which are the ones that we should promote to help sperm health. We've also looked at sperm themselves. We've looked at the quality of sperm in unexplained infertility. We're looking at sperm sorting techniques. Are there better ones than are used in the clinics at the moment? And the one that excites me most is how much repair can be done by oocytes, because we know that the oocyte can repair a lot of sperm DNA damage post-fertilization. Don't want to talk too much about the exact test, but just to say that this is a typical plot for an infertile man. And with the COMET test, we can see a lot of different results. We pick out three. One is the average damage across the whole range of sperm that we've looked at. One is the LCS, or the low COMET score. Those are the green sperm, or the good quality sperm, the ones that a fertile man would have. And we can also look at the high COMET score, and those are the really bad ones, usually badly damaged by oxidative stress, but they also have the capacity to damage other sperm that are adjacent to them. All the tests in our lab are generated to ISO 13485 certification standard, and we're also working towards an ISO 15189. And the interpretation of our results and the management of the client is performed by the referring clinician. Now, over the last couple of years, it's been very exciting because a number of international guidelines have changed to include DNA testing and also to include examination of the man himself, because we've thought of the man as just being a sperm donor for so long, and we haven't actually looked at the man himself and how an improvement in his health can improve his sperm health. So the European Association of Urology has changed its guidelines to say that, as well as a semen analysis, it's sometimes the case that more complex testing, such as DNA testing, is required. The ESHRA guidelines, the European guidelines for recurrent pregnancy loss, when they came out in 2017, there was it was suggested that it could be considered looking at sperm DNA for explanatory purposes. Now, in the latest one, which has come out in 2022, which is out now with stakeholders, we're saying that DNA testing should be done on these male partners of women who've had recurrent pregnancy loss. So that's a huge step forward as well. The American Urology Society and the American Society for Reproductive Medicine have both changed their guidelines, again, talking about the male partner being evaluated, not by a gynecologist this time, but by a male reproductive expert, and consideration being given to DNA fragmentation. And of course, one of the most exciting things that happened last year in 2021, and again, Professor Lamb was very much involved in writing the WHO laboratory manual for the examination and processing of human semen. And in this, an extended examination, now it is recommended that sperm DNA fragmentation can be measured there. It used to be just part of a research tool, and now it's part of an extended examination. So if we get to this wonderful person who's speaking for us tonight, if I were to say she was amazing, I would be underestimating what she has achieved. She's one of the first people who defined the genetic basis of male infertility. And as well as doing all the empirical research, she is well recognized for her capacity to get alongside people, to mentor them, to develop clinical scientist relationship. Look at all the things that she has achieved. She's been distinguished andrologist award, distinguished researcher award from the ASRM, which is no mean feat. Distinguished Mentor Award and Mentoring Award for the Society of Women in Urology, so she hasn't forgotten about her own gender as she's climbed the ladder of success. The Endocrine Society Laureate Award for Mentoring, the Cavosi Family Teacher Award from the American Society of Reproductive Medicine. She's actually a former president of the ASRM. I think she was saying earlier that that is something like 12,000 members. And she's been involved in other societies. And the one at the bottom is the most exciting of all. She's been continually funded by NIH for 40 years for her research in andrology and urology. So that is a CV second to none. So I'm going to hand over now to Professor Dori Lamb and allow her to talk to us about the changing face of male fertility. And I am sure we're really going to enjoy what we hear. Thank you very much, Dori.
Thank you, Sheena. I'm blushing.
Good.
Very kind. Okay, so I think you can see the slides now. Thank you so much, Sheena and Eileen, for the invitation to come here to speak to you all today. And as Sheena said, I have a long history of work in reproductive medicine. I not only did most of my research in looking at male infertility, but amazingly enough, I also worked on early pregnancy, trying to understand the fetus as a fetal allograft, and also on implantation and uterine function, again, in early pregnancy. So I've kind of worn both hats in terms of working in the male and working on female reproductive aspects. So these are my disclosures. None of them are relevant to the topic of this talk. I would say that obviously as a Secretary Treasurer for the American Board of Bioanalysts, where we work under deemed status by the US government, we set the tone for the requirements for specialists in andrology and in embryology in the United States. And so I also use a lot of my clinical laboratory expertise as well. And so today what I'm going to talk to you about is really kind of a little mixture of some of the clinical andrology as well as some research aspects that I think are kind of the future of the field. And I think probably you all know Chris Barrett, who co-authored a paper with me that came out a few months ago in Fertility and Sterility. This was part of 4 individual manuscripts that were all submitted together, talking about WHO 6, because Chris and I were both authors, some of the editorial board of that, who wrote the new 6th edition of the manual for the WHO laboratory manual for the examination and processing of human semen. But it was really a joint effort between the two of us, as well as all the other members of our editorial board. But I thought that I would sort of couch some of my comments today in light of the chapter or the journal article that we both wrote for fertility and sterility, looking sort of towards the future, where we are now and what's going to be needed in the future, and a little bit also about WHO 6 and some of the very positive things that we hope to come out of that. So, I think that we're all very familiar that the routine semen analysis, the history and the physical, are the cornerstone of the evaluation of male reproductive health. And it usually, in many instances, certainly in ART today, the semen analysis is the only test that's done on the male of a couple seeking treatment for infertility. And, despite the fact that this test is so widely used, it is so poorly performed. And we know this because there's a huge amount of data that come from reports all over the world, where, especially in areas where there are very good proficiency testing strategies, right, which use external quality control samples to look at the accuracy and precision of semen analysis reports. And independent of whatever country you live in, it shows that there is huge variability in the results of the basic semen analysis. And ideally, we should all get the exact same answer, or, certainly be within the limits that are acceptable for doing a semen analysis. And it's very clear to us that much of this comes from a lack of adherence to established methods. And we know this. There was a paper by Dr. Nieschlag a few years ago where he actually looked at how well the WHO standards at that time, the guidelines, which was the 5th edition, were being followed. And there were all these reports in the literature where people said that they were following WHO 5, and they weren't following WHO 5, and for many, many reasons, counting chambers, the way they counted, the lines they counted in the chambers. Some of it represents also not just following those guidelines, but poor training and poor assessment of competency and criteria for competency to perform a semen analysis. And again, the use of all these different methods to perform a semen analysis. And I'd also mention, for those of you on the more clinical side, that in WHO 6, CASA was actually moved into the research area rather than something that should be used clinically. And the reason for that was because there is such huge variation between different instruments from the same manufacturer, same model. And then, of course, when you try to compare results between different CASA system manufacturers, you have different results. And all of these results are different again from are the results in a routine semen analysis done manually. And so it's not to say that there are not settings where this is a very useful tool to be using. And there's obviously great opportunities with all of our computer technologies now for improving this. But, it was felt that from a clinical diagnostic perspective, that it was not sufficiently accurate and precise relative to the gold standard, right, which would be the semen analysis performed manually. But because of all these differences in how people perform the assay, it's impossible to compare the results from one lab to another. And so that becomes a big problem. And so, our goal in WHO 6 was really to try and provide clear, simplified methods that would address the reproducibility crisis, and then, help to improve kind of the overall quality of semen testing throughout the world. And we felt that by making these protocols very simple to follow, that then training could also be facilitated, again, improving competency for doing a manual semen analysis. So why do we want to do this? And why is this routine semen analysis so critical for the evaluation of male fertility? So I'm showing you here kind of a schematic of data that was originally published by Guzak in the New England Journal of Medicine in 2001. The paper is highly quoted. I'm sure many of you have read it. And so this was from the NIH-funded Reproductive Medicine Network in the United States, where they looked at a very large number of patients, and they divided them into patients who were proven fertile, patients who were infertile, and they asked very simple questions. And here I'm showing you their study on what is the distribution of sperm concentration in fertile men. And so you can see many things from this kind of chart. The first one is that, semen parameters are not normally distributed. And there is a wide range of sperm concentrations when reported as a million per milliliter that individual men have in their ejaculate. But I think it's also noteworthy that, can you all see my arrow here? Okay, good. So I think it's also noteworthy that, a large number of proven fertile men are oligozoospermic. And some of them have severe oligozoospermia. And in fact, the lowest concentration of sperm, or actually amount of sperm in a semen sample that was able to achieve a pregnancy was reported by Rebecca Sokol many years ago. And the man had 50,000 sperm total in his ejaculate. And he was able to naturally conceive a child, even though he was thought to be infertile based on his sperm count. And paternity was documented. So it was actually not the neighbor that did it. It was from this man's sperm. And so even men with very low sperm concentrations in their ejaculate can be fertile with no assisted reproductive technologies applied if the sperm work well. So then they looked to say, what is the distribution if we compare fertile versus infertile men? And essentially, the values that were reported over again, the same sperm concentration range were virtually equivalent for men who were considered to be fertile and men, and they were, right? They had proven fertility, but the infertile men were considered to be infertile, and yet their semen parameters were really indistinguishable. And, so this raised a huge quandary, right? Because there is no way to predict a man's fertility based on his sperm concentration. Now, they also pointed out in this paper was that the higher the number of different types of abnormal semen parameters that you had, the higher the probability or the chance of infertility. So here, they're plotting the number of abnormal parameters as compared to the odds ratio of infertility. And so, for example, if we think about types of abnormal semen parameters, for example, for the male who has just a asthenozoospermia, right? He has an increased odds ratio, although very slim, as compared to no abnormal semen parameters. But if then he has both asthenozoospermia and poor morphology, teratozoospermia, then he has an increased odds ratio. And similarly, if we compared, what did I miss? Let's see, motility, morphology, and count. So then again, they would have a higher odds ratio of infertility, but on an individual patient basis, you could advise them of their chance of infertility, but you could not describe them as fertile versus infertile based on simply looking at the semen parameters, because you could not do that. You couldn't say for an individual patient whether or not they were fertile or infertile. It was only the population overall that led to this data. So, what does the semen analysis tell us? It's telling us a lot about testicular function, sperm production by the testis, the genital tract secretions, the patency of the genital tract, but it is not a test of fertility. And this is because there are so many other things that the sperm has to do other than simply exist, that we can't measure in a routine semen analysis. So the ability of sperm to undergo sperm capacitation, to bind to the zone of pellucid, to penetrate the egg, and then undergo all the steps of nuclear decondensation required to fertilize the egg. So at one time, some of the sperm function tests were very popular. I actually worked a lot in this area as well. These tests are not widely used anymore because everyone is simply put for getting treated with intracytoplasmic sperm injection with ICSI, with IVF. And so nobody really pays attention to this, although some of these tests are actually quite useful to predict patient outcomes with these assisted reproductive technologies. So, we tried to think very hard then about what was going to advance the field and what were the things that were needed and kind of looking towards the future. One of the things that we really thought was very powerful is that in today's, environment where, we have access to meet with people all over the world. I'm in California right now, you're all in Ireland. And, what an advantage this is for all of us to work together as colleagues in our discipline, and really address, formulate key questions that need to be asked in the field. And, we could all write a book here on the needs for, trying to learn more about male infertility. And really address these questions and do it in a global perspective to achieve, I think really optimal results, assuming we can all run these tests and analyses in a standardized, cohesive way. And of course, that allows us to gain this information very quickly as well. And I think that one of the things we were also very taken with are some of the new artificial intelligence methods that use machine learning. I actually have some of the very early papers on this in any medical application of artificial intelligence with Craig Niederberger, who is my third fellow, now chairman at UIC, a urologist. I thought I was quite the computer jock. He far surpassed me in those skills, but we actually wrote our own program for performing what's now called machine learning or neural networks to try to look at a variety of different types of male infertility needs, including some of these to try and pick the ideal sperm. And of course, the technology has increased exponentially since then. But, we now have very interesting papers published that are looking at using these machine learning techniques where they're watching, various aspects of sperm morphology, sperm motility, all the things that we might measure with a CASA system, and try to use these technologies to identify new biomarkers that could help us to predict key sperm functions. And so some of these are parameters such as actually measuring the intracellular pH of the sperm themselves to predict capacitation, or to try to predict conventional fertilization success in men with normal semen parameters, or even to pick the optimal sperm for ICSI in a non-invasive way. And to do that, again, based on a whole variety of different types of parameters, not ones that humans can't discern, perhaps, some of them they don't, but when you have these complex patterns that need to be recognized, maybe to identify that ideal sperm, the computer is quite good and does a great job at that, and kind of takes away technician skills and so on from maybe picking the best sperm for ICSI. for example. So we were very taken with this, and we thought that this was going to be an area that's going to be hugely expanded, both on the research and commercial sides, as you can imagine. And, we also felt that there's a huge need for development of new advanced tests, extended assays. And again, I was the primary author for chapter 3, which Sheena had mentioned, of the WHO manual. Obviously those were the tests that are ready for prime time now, or thought to be, clinically validated and giving a diagnostic answer that would allow treatment changes or, changes in the therapy choices of couples for trying to achieve parenthood. And one of the things that's really revolutionized I think our ability to maybe change the way we are diagnosing infertile men is the advent of next generation sequencing. These include technologies such as whole exome and whole genome sequencing. But if we look, and these are really ready for prime time many times, but there are other omic technologies that I think certainly in the next 10 years will become far more commonly used. Array comparative genomic hybridization is really quite standard in the clinical lab, so that moves up more with the whole exome and whole genome sequencing, but also the other omics, right? So transcriptomics, proteomics, metabolomics. Metabolomics is, I think, really the wild card to be watching, because that measures the end result of the entire system rather than looking at, for us, say in my field, looking at the DNA, right, which is the starting point rather than the end result. And crazy things, where they're taking, say, a contact lens and using it then, taking it out, doing a metabolomic analysis of it to be able to identify malignancies, that are not even imageable yet, but based again on these various changes that occur in the system itself of the individual, like the entire body, rather than just focusing on the gene, the protein, or so on. So I think, there's great excitement for the future in terms of these diagnostic advances. But, there certainly has been an exponential increase in the research reports on the molecular, cellular, and again, these system defects that are present also in a wide variety of male reproductive deficiencies. And I want to give you a couple of examples of this, all of them that relate to male fertility. So, I want to talk some about genetic and genomic tests. And I think that you're all quite familiar with numerical and structural chromosomal defects, mostly clinically measured by a karyotype, as well as gene deletions and duplications and mutations. And then there could also be obviously epigenetic mutations that people are, again, actively trying to commercialize and develop clinical diagnostic tests using all of these types of technologies. But I'm going to start here by talking about genitourinary birth defects, which is one of the things that I work on as a research perspective. And, these are important for us. These are the most common birth defects. And I have the incidence for you shown here. And very interestingly, you can see that they're incredibly common. Cryptorchidism, for example, is one of the most common causes of male infertility, even after an orchiopexy was performed, right, attempting to maintain or restore spermatogenesis. But, many of these others can be associated with infertility, certainly ambiguous genitalia, or if you have a condition such as gonadal dysgenesis or whatever, these are causes less common of male infertility. And even having renal agenesis, right, can be associated with male infertility, because many of these patients have unilateral renal agenesis, but they have also bilateral absence of the vas deferens or unilateral absence of the vas deferens. Again, a plumbing problem that prevents the sperm from transiting through the genital tract, and those men would also be infertile. And it's very interesting because these GU anomalies frequently come in groups, right? So a patient with hypospadias could also have renal agenesis that might not be picked up unless he had imaging. Or there can be other developmental problems beyond just the genitourinary tract. And this is because they all have a common embryonic origin, which is the intermediate mesoderm. And so my lab actually, I would say, pioneered the use of array comparative genomic hybridization, which was invented at Baylor College of Medicine, where I used to work. And I gave them DNA samples from my Y chromosome microdeleted patients that we were diagnosing using a multiplex PCR assay for them to use as a positive control when they were inventing the technology of doing array comparative genomic hybridization, where you have an array-based technology. First, they had BACs and PACs on the array. So these are like artificial chromosome segments, bacterial artificial chromosome segments. And now they are oligonucleotides spanning the entire genome. And what this test measures is what's called copy number variations that result in gene dosage changes. And these copy number variations are microdeletions or microduplications too small to see on a karyotype. So the Y chromosome microdeletions were the first that were really identified and studied in any great detail. But we know now that they can be on the sex chromosomes, the autosomes. Any chromosome can have some of these microdeletions or microduplications. And so normally, we know that all of us, get our genomes from our mother and our father, right? So we should have one copy of a gene from our father, one from our mother. 2 copies would be normal on an autosome, for example. But if during homologous recombination, when the chromosomes come together, and they synapse, right, and they cross over and then they segregate, if there's a problem with that segregation in a small region on the chromosome, you can end up with essentially 2 copies of that little region of the chromosome, right? For example, here, if this was the sperm that was then contributing to the offspring, the 2 copies from the sperm plus the 1 copy from the mother would end up with having 3 copies, which is too much of that gene being present, too much of that protein being present. So it's a way of changing gene expression based on how many copies of the gene you have, not through the promoter, anything particular with turning on a gene to increase expression of the gene. So it's very much DNA based, but it results in increased levels of protein. And then, again, with that combination, right, the 2 copies went to this sperm over here. When no copies went to the other sperm, if that was the sperm that fertilized the egg, again, you would have 1 copy from the mother, none from the father. And again, that's abnormal because you should have 2 copies of this to get the proper amount of whatever protein that gene is encoding, if it is dosage sensitive, to affect the function of the organism. And so again, microdeletions, we all know them from Y chromosome microdeletions, but again, remember they can be on autosomes or other regions of the sex chromosomes as well, or microduplications. So sort of too much or too little of a good thing is a bad thing when it comes to lots of aspects of adult function as well as development. And we were focused on development. And what we found was that about 1/4 of all the non-syndromic males born with genitourinary birth defects, and our patients were basically hypospadias and or cryptorchidism, can have autosomal or sex chromosomal regions, microdeletions or microduplications, and they all clustered by what type of birth defect they had at specific regions of the chromosome. And that told us that there were hotspots encoding genes that were required for normal development. And those studies have moved on now to not only look at these gene dosage changes, right? And again, this is just giving you the schematic of what the deleted region, is, and this is a big deletion, or a duplicated region would look like on the chromosome. And, if there's a gene that's impactful in terms of genitourinary development, if you have also damaging mutations, right, you can get the same phenotype being seen. And, it can affect reproductive system development and function, but it also, remember I told you that it can affect other body parts. So fetal, childhood, adolescent development and or function or other organs in the body. And what's so interesting is that the genes which were shown to be causative for these genitourinary birth defects so far, affect major signaling pathways, for example, androgen receptor function in ways never before considered, that result not only in these genitourinary birth defects, but in some cases also spermatogenic failure in the adult. So again, these would be the patients who have cryptorchid at birth, had the testis brought down into the scrotum during, early childhood, infanthood, but they still have spermatogenic failure. And that's true for about 65% of infertile, of cryptorchid men, that they are infertile with spermatogenic failure, regardless of whether they have unilateral or bilateral cryptorchidism. So it's a major cause of male infertility. And, as I told you, in addition to these candidate genes that have since been functionally found to be causative of these human male birth defects, there's nearly 3,000 genes which have been identified and associated with human hypospadias and with cryptorchidism. But first of all, these patients are never diagnosed, because they don't order whole exome sequencing, which would identify many of these defects. But it's important to know that these defects are causing other problems. And so the urologist looks at the baby with the birth defect. He doesn't worry, and I'm talking about the pediatric one, doesn't think about the fact, cryptorchid, good chance that this child is going to be, have spermatogenic failure when he grows up. He just thinks, very easy for me to put these testes down into the scrotum, simple operation. And even, unless you have very, severe hypospadias, a lot of that is very easy clinically to fix. So they never pay attention to look at the whole baby. And yet we know from these studies that I'm telling you that of the 15 genes that we've identified in the past 10 years, forward genetics, again, as I described, in patients with, cryptorchidism, hypospadias, as well as some of the upper tract defects and testicular defects, that they're syndromic, which means that even though they were diagnosed as that hypospadias or cryptorchidism was their only birth defect, they have other problems that nobody's looked at yet. So some of them could have cognitive dysfunction, particularly in the cryptorchidism patients, like head and facial abnormalities, cardiac defects, skeletal malformations, hearing, vision problems, CACUT, which again are all the renal problems and upper tract problems of the urinary system. But these things are not happening in isolation. And again, these patients, some of them, would greatly benefit from an early diagnosis of these syndromic problems rather than finding this out as they mature. And they never connect the two, that the GU birth defect has anything to do with, with say the vision problem or the hearing problem. It's just not thought of. And so, I think that this is like a good example where, no one really recognized the problem of genitourinary birth defects and spermatogenic failure. And it's important that we know more, because from a diagnostic perspective, you could even do this prenatally, right? To identify these abnormalities. And it is important to remember that of those 3,000 genes that I told you are associated, with these birth defects and with spermatogenic failure, a lot of them have not undergone the rigorous testing that you need for clinical diagnostics to really prove that it is causative to the birth defect and the spermatogenic failure. And it's important also to know the mechanism of action, not just that it's causative, but you need to know the mechanism of action, because then you can certainly understand the patient phenotype. And sometimes, you could use drug discovery, perhaps for new medical therapies that might, ameliorate the need to do some of these, surgical procedures. But there's no doubt that the data will be informative and useful in the diagnosis of these patients, because right now the causes remain, clinically unknown usually. Now I want to just switch gears very quickly, going along the same sort of line of thinking, and just tell you about genetic anomalies that impact the sperm morphology, not sperm production, as we were talking about for the GU birth defects, as well as their function. And, I think there's a strong sense among many investigators that, the routine semen analysis is probably going to be eventually not replaced in whole, certainly augmented, but perhaps much of the work be done genetically to diagnose the patients who are infertile. So I want to give you two examples. One has to do with teratozoospermia, and then the other examples have to do with asthenozoospermia. These are not the only ones possible, but just, in the interest of time, just to tell you a quick story about these. So we're going to start with teratozoospermia. So, we're all very familiar with this in andrology. And in particular, the studies that I'm going to show you first have to do with globozoospermia, which is round-headed sperm. This is when the acrosome is either absent, as we see right here, or misplaced, atrophied, as we see down here. And this is by confocal microscopy. You see the same information over here. But this is a big problem because in the absence of having the acrosome, the sperm cannot penetrate the egg and achieve fertilization on its own. So, these individuals usually immediately go for IVF, and many of them do not fare well with ICSI. Sometimes the egg is not activated with the injection, or even with doing calcium activation to allow fertilization to proceed. And even if it does, the pregnancies are not as, you have a lower live birth rate using globozoospermic sperm. And so, again, with kind of the genetic revolution that's occurred, we are beginning to learn a lot about all of the different genes that cause globozoospermia, a whole variety of these, and the list keeps exploding exponentially. And what they do is they prevent the acrosome from actually, developing and forming. They prevent the process of the acrosome forming to actually then have properly formed mature sperm. The two important genes to know, which are the most commonly ones affected, are SPATA16, which is spermatogenesis associated protein 16, highly expressed in the human testis, and damaging mutations in this gene are associated and found in a significant percentage of globozoospermic men. The second gene is DPY19L2, not catchy in terms of a name, but here you can have either the gene dosage changes that I told you about, copy number variations, or you can have damaging mutations, more common in some geographic and ethnic regions. But again, you end up with a high percentage of abnormal sperm, and both of these are common defects that underlie globozoospermia. So you could say, well, why do I need to know this? Well, when couples have, either the mutations or the CNVs of these genes, while they have normal sperm density, with globospermia, the likelihood of achieving a pregnancy and a live birth, even with oocyte activation for these two gene defects, is much lower than occurs with IVF ICSI with sperm from an intact normal acrosome. And, couples need to be advised, that their likelihood of having a healthy baby at the end is quite a bit lower than if they weren't globozoospermic. And so, and again, couples may make an educated decision on that. And I'm talking in both of these cases on complete globozoospermia. Sometimes we see kind of a mosaic mixture of that. This would be the complete forms. Another type of teratozoospermia gene defect, which has been shown, is due to aurora kinase C mutations. And again, we see a very specific defect in spermatogenesis, and that is that when you have either absent or dysfunctional aurora kinase C, you don't get the proper steps performed to result in proper chromosome segregation during meiosis, and you end up with a blocked cytokinesis. So you get no cell division during meiosis, and what you result in is, rather than ending up with, say, 4 sperm, as you should from this single metaphase cell, you end up with 1 sperm with tetraploid head, where you have sort of a large multinucleated head, many times with 4 tails. And so, very easy to see on a routine semen analysis. But again, when you have this type of defect, macrozoospermia, the big head, this is one of the most common causes of this. It's particularly common in men of European and Northern African origin. And again, this changes treatment, because for these men, ICSI-IVF is not recommended, because most of them cannot achieve a normal pregnancy, again, because of various types of having multinucleated sperm heads being injected. And then finally, we'll just touch quickly on asthenozoospermia. And, many places now are using multiple morphologic anomalies of the sperm flagella to describe a variety of different types of flagella defects in sperm. And so there's been a huge interest and huge advances in identifying all types of proteins that encode every single structure that we see when we look at cross sections of the sperm flagella, and we look at them, say, by an electron microscopy. But there are a lot of proteins which are involved in getting the sperm to not only form properly, flagella, not only to form properly, but to beat properly. And the most common mutations found in sperm with poor motility are mutations in the dynein axonemal heavy chain 1 DNA H1 protein. Here, this is the cilia and flagella associated protein 44 and 43. And then there are some other mutations found in other proteins that, again, encode the structures that form, say, the microtubule apparatus, right? The fibrous sheath of the sperm, and defects in all of these different structures that are found in the flagella result in a variety of different types of motility defects, and in some cases, morphologic defects as well. And, primary ciliary dyskinesia is a well-known cause of motility, total immotility. But there has been an explosion, and this is considered to be a different category of defects, although you'll see common genes as well. And it's characterized by airway disease, male infertility, right, due to totally immotile sperm, laterality defects, which is situs inversus. And here you see a dual loss of the inner dynein arms and the outer dynein arms that power the cilia or flagella. So you have totally immotile sperm. And this was easier to follow because they had affected families, the men and the women affected. So they were able to do pedigrees and whole exome and Sanger sequencing to identify even biallelic mutations in them. And so today, clinically, patients with this type of immotile sperm would be tested with panels in clinical genetics labs of over 90 different gene defects. And again, remember that these defects are affecting the axoneme, the inner and outer dynein arms, the regulatory complex, the central microtubule pair, nexin links, and laterality. And but on top of that, there are almost 1,000 other genes, which again are in the process of being validated and causative for seeing these types of motility defects. So again, we're looking at big data types analyses, just even for, say, a motility defect. But the fact remains that these individuals with poor motility sperm have a variety of different types of genetic defects, which contribute to what we see in a routine semen analysis. So, I guess I hope I've convinced you that, certainly that the semen analysis has to be performed very accurately. And that is really going to set the stage for advanced protocols, where we can do advanced clinical trials of all these other aspects of types of testing that are really going to improve our ability to understand the causes and perhaps develop new treatments for a variety of different types of male infertility. Obviously the studies have to be focused at fulfilling the evidence gaps in the diagnosis and treatment in order to ultimately improve clinical care. And I think that the power of the genomic revolution, and then we can add on to that all the others, transcriptomics, metabolomics, all of these are further in the future for male infertility, but they're really going to allow quantum leaps, I think, in improved diagnosis and andrology. And by being able to identify these precise etiologies, comorbidities, I gave you the examples of the GU birth defects causing cryptorchidism with spermatogenic failure, and potentially medically based treatments for men with reproductive failure could hopefully improve their fertility potential as well as their overall health. So, I want to thank you all very much for inviting me to talk, I'm sure, hopefully we're going to have a bunch of great questions. I look forward to meeting you all in January when you all attend the meeting in Dublin. So Belfast, is it Belfast?
Yes, Belfast.
Belfast.
Yeah.
So I haven't been to Ireland yet. So, but I look forward to meeting you.
Oh, thank you so much. That was really amazing. An awful lot of information there. The people that we have listening tonight are from all over the world. We've got quite a number of different countries, so it's not just people from Belfast, or even people from the UK. So the information that you have given tonight will be spread globally. So that's pretty good.
Excellent.
What I would like to ask you is a strategic question, in the sense that who is going to drive this? Who is going to drive the fact that we know so much, we have so much science, we're aware of so many things, but actually they don't take that quantum leap into the clinic? Because I'm not sure that I ever heard of a person with macrozoospermia being recommended not to have IVF or ICSI. It was just, let's do it and see what happens. Or even with globozoospermia, which is a milder problem.
Sure.
So what do you think?
Well, so, first of all, just on the end part of that question, I think it's recommended, but not told, right? Because obviously couples should have the freedom to make their reproductive decisions. But I just think it gives them an educated perspective that the likelihood of having a healthy child at the end, which is everyone's goal, could be far less or zero. And, it's financially and emotionally so draining, right, on their couple resources, if you will, both personal and money-wise, that, it might help in their decision making. And there's no right answer, right? Obviously. I think that, though, the question you asked, Sheena, is so timely about getting this into the clinic and getting this into reality, right? It's one of the biggest challenges in medicine. And I can tell you that, for example, in oncology, as you know, I also had a huge history doing all the PSA testing, all the tests, you know, for bladder cancer, all these other tests, and then also trying to understand kind of like, when do you biopsy a man, when do you not biopsy for prostate cancer, for example. And because it's not overdiagnosis, it's overtreatment that's the issue, right? And I get called all the time by urologists who get a molecular test run, you know, that gives probabilities, right, of you know, relative risk for finding prostate cancer on a biopsy, or aggressiveness of a tumor. And they have no clue. It says normal or abnormal, but then, you know, all of the results that are there, they don't have any clue what that is. And it's the same for when many of the surgeons say get a karyotype analysis. And, you know, there's like really bad karyotypes and not, but they have no way of distinguishing. And some of them don't even particularly know what they're looking at, you know. And so some of it obviously is education, but it seems that I think in today's society, people think that, oh, they don't need to memorize facts and they don't need to have the huge foundation of knowledge in the field, right? Because they can look it up, but then maybe they look it up and it's still not informative because they're lacking that foundation that they should have gotten in medical school or whatever. So it is a challenge, and I'm not sure that there are easy answers. And you know, I know even from teaching some of the postgrad courses where we were very basic in terms of the and this was for urologists, the information given, there were REIs there as well. But it was very clear that they didn't pay attention because you could see them all on their phones and everything else. But they also didn't learn it, you know, even though it would have helped them tremendously in terms of, you know, some of their practice, right? So do you know? I mean, it's a challenge.
I don't know. I mean, I certainly take your point entirely that a very good basic foundation is the best way to move forward, because you can't always be looking up a book, or you can't always be looking on your phone to get the answer when you're talking to a patient. It should be something that you learned previously. We have a question here from Gulam Badahur, and he says, we know that the non-discriminate use of ICSI does not improve pregnancy outcomes over IVF. Going further, presumably many thousands of ICSI babies would need to be looked at to show whether the random choice of sperm was problematic and impractical. Now that's a tricky one, bearing in mind the way life has gone, both in Eastern Europe and also in the States, in terms of ICSI being the choice rather than IVF.
Right. And, you know, and I actually used to lecture on this a lot because we were doing ICSI. We were too gentle. You know, I'm saying before John Pero, right, you know, made his little mistake there and was a little rough there with the egg. But, because I never would have been that rough, but once we learned the trick, we were set to go. And it was incredible to me because, you know, it was designed for severe male infertility, and yet it's so widely used. And it's even with DNA fragmentation, right, where people say, oh, you know, we should, you were asking earlier, take sperm from the testis, right, less damaged DNA or whatever. But you don't know with the individual sperm that you pick up, whether that's a healthy sperm, a good sperm. And, you know, in the embryology world, and if anybody's an embryologist, maybe you can correct me, but in many IVF groups, you know, anything that sort of looks like a sperm is considered to be a healthy sperm. And all they need is the DNA, more or less, right, to be able to achieve a pregnancy. You know, we don't honestly know any of that. And I think that, I think that it's absolutely correct that indiscriminate use of this, you know, bypasses all of nature's barriers to fertilization by defective gametes, you know, on both sides, right, the egg and the sperm. And we don't totally know the whether or not that's a good thing to do. And I think that the reason that the IVF and ICSI results are also so similar is that most of the people, there are so many people getting ICSI and IVF that don't need it, right? They're kind of diluting out what would be the more severe male and female factors, right? In terms of looking at the safety of these procedures. And as you know, it's very difficult to do those studies that are well designed, that couples are going to agree to. And, you know, we could probably have a whole talk on that.
Oh, we absolutely could.
Yeah, but I agree completely. I mean, I think it's a big issue in the field. And, you know, nature kind of said, keep these bad genes out of the gene pool. And instead, you know, we're just bypassing that.
Yeah.
So, yeah.
I was shocked at the statistic of 1 in 30 children with cryptorchidism. And I'm just wondering if you
yes.
Can you tell us a little bit more about what potential causes there are? I mean, why? I mean, as far as I understand, there's a significant increase over the last couple of decades. Is that correct?
Well, so the increase is not agreed upon. You know, and I know the studies on the, you know, sort of the endocrine disruptors, the environmental agents, and you know, that I can't comment on because in humans, we don't have good data on a cause and effect relationship to be causing that. And when you look at sort of the numbers of that, you know, I think that one of the problems is that so many women are put on progestins, right early on in their pregnancy. And sometimes it's done routinely, right, by some of these ART groups, you know, hoping to, you know, progesterone's the hormone of pregnancy, hoping to, you know, have an optimal then sort of environment. And, you know, but we also know that these exposures to that result in cryptorchidism and hypospadias, more so even hypospadias. But I think that, you know, there's other things going on that could potentially impact the frequency. The numbers I'm showing you are actually the conservative ones. It's usually about 6% of full-term newborns. Then in the first 6 months, for some of those newborns, about half of them, the testis will descend. Some of them have retractile testes as well. But those numbers are very solid. They're pretty much worldwide. I was amazed also, and also that no one had any clue. I can tell you that all of the proteins the genes that we identified with these copy number variations, we are accounting now for about, what would my number be? About 18% of, you know, cryptorchidism, hypospadias, right, and so on, from the defects that we've identified. And we have high impact papers in Nature Medicine, PNAS, you know, and so on these genes. And we go the whole gamut, right, to prove that it's beyond an association. But all of these genes affect signaling pathways in ways we never knew. And so for example, one of the proteins, when overexpressed, impacts the androgen receptor transport with the ligand into the nucleus. So it's not that you get none going in, but you get a blunted androgen, you know, effect. And then estrogen receptor is potentiated. So even though the baby's hormones haven't changed, right, you've kind of disrupted the yin yang between, because, you know, both estrogens and androgens are important, right? But it's not a hormonal problem, it's a receptor problem. You know, there's some that affect Wnt signaling, right? Like for the developmental processes, some that affect RET signaling, cell polarity. I mean, there's a whole slew of different like processes that are affected. And quite honestly, we had to think very creatively, because, you know, we were expecting to find like androgen receptor co-activators or co-repressors being like major players, and that wasn't it. And we just let the data talk to us, and we had to think very creatively about how we thought we could end up with the birth defect. And, you know, we kind of learned that like we couldn't have our preconceived notions, right? And instead we had to just, you know,
to speak
have
and
fun.
yeah.
Yeah.
Well, I think we've run out of time, Dory, but I must say I really would like to thank you for everything that you've shared with us tonight. There are lots of messages coming in saying thank you for your great talk. Can't wait until gene testing replaces and supports analysis, and also people who are so delighted to hear that some really, really good research is being done, because we so seldom hear about it, and certainly those of us who are working much more on the clinical side of it are not exposed to the knowledge of like yours. So I'll hand you back to Eileen now, but thanks so much.
Oh, it's my pleasure. And thank you so much for inviting me. Whoops. Yeah. And I appreciate it very much. And I see John Sullivan there, who's one of my pals. So yeah.
Yeah, well, John has come back to Ireland now. So
yes, he has.
We'll be working a lot with him in the future.
Yeah. And Chris Barrett. Excellent. Excellent.
Yeah, thanks again, Dory. And I think you mentioned Chris Barrett earlier, so some familiar faces that are coming up for our next events. So hopefully all of you online can join us for that. So the next webinar takes place on Tuesday, the 7th of June, where we're joined by Professor Christopher Barrett, who's a global leader in male fertility and was appointed to the editorial board, as you heard earlier, of the World Health Organization for the development of a new semen analysis manual. So he recently published the global WHO guidelines for semen reference ranges, and will be discussing why a global approach is needed to address the policy, research and social challenges of the male reproductive health. So very much backing up what you've just talked about as well. And then on the 2nd of August, we'll be joined by Dr. John Sullivan, who's the Senior Registrar in Urology, now just recently at St. James's Hospital in Dublin, and Fellow of the Royal College of Surgeons. So Dr. Sullivan joins us to explore what advances are needed to provide equality of care for men and women wanting to start a family. So if you haven't already registered for the events, please do so on the events page on our website. So that just leads me to say thank you again for all of you for joining. If we could ask you to provide feedback at the end of the event, that'd be really appreciated. And our contact details are on screen if you need to get in touch with us at all. And hopefully we'll see you at the next webinar on June 7th. So thank you again, Professor Lamb. It's been an absolute honor to have you join us today. And thanks to Sheena as well. And thanks all of you for joining, and enjoy the rest of your day.